So what are some of the systems we can use? Obviously, these are all perfusate systems. There's three main ones that we're going to talk about today, either crystalloid-based, whole blood perfusion, or an erythrocyte preparation. So when we talk about crystalloid perfusions, we're mainly talking about Krebs-Hanslett buffer, or KHB, and it's the most widely used perfusion buffer associated with Langendorff perfusion. It contains those ions that are going to mimic the plasma content, so things like sodium chloride, magnesium sulfate, potassium phosphate, sodium bicarb, and calcium chloride. The buffer needs to be bubbled, importantly, with not just oxygen at 95%, but also some carbon dioxide. And it's the carbon dioxide that is necessary to maintain that physiological pH of pH 7.4. It is important to note that the physiological levels of these ions needs to be considered when developing an experimental protocol. Most of the components of the KHB will actually be at supraphysiological levels, for example, calcium and potassium, and we need to make this a consideration before we actually start our experiments. Obviously, we're providing an exogenous carbon source for cardiac metabolism. The most popular choice, of course, being glucose. However, this is too supplemented at a supraphysiological level, and depending on the biology in question, it can influence functionality, especially when things like glycolysis is a contributing factor. Pyruvate can also be used as a carbon source, as well as free fatty acids. However, there can be some solubility and foaming issues with these last two carbon sources. Lastly, given that Krebs-Hanslett buffer is a protein-free system, the oncotic pressure is significantly lower than physiological levels, increasing the total tissue water, which can effectively simulate an edemic situation. This is further exacerbated by the supraphysiological flow rates required. In the absence of hemoglobin, the oxygen carrying capacity to the vascular bed can also be decreased relatively. Even in light of these supposed limitations, the crystalloid perfusion system does remain popular, mainly because of the low cost associated and the reproducibility of the production of the buffer systems. Secondly, whole blood perfusion systems. The advantage that this system offers is the ability to maintain a perfusion system that is nearly identical to the physiological setting. By setting up whole blood perfusion, it can be significantly more challenging by comparison to those crystalloid perfusion systems that I just mentioned. The main drawback of this system is that it originally suffered from an incapacity to reoxygenate the blood after ejection from the ex vivo coronary circulation. To circumvent such an issue, a donor animal can be used to essentially reoxygenate the blood. This recirculation also means that the blood must be inline filtered, which can pose additional issues. Flow rates in the whole blood perfusion system are significantly lower. However, there is a significant risk of hemolysis or blood cell breakdown, and the humoral effects of the donor animal can have an influence on the ex vivo heart. Conversely, when we consider that the perfusate coming back from the ex vivo heart may contain contaminants, this can have an influence on the donor animal. And these are considerations that need to be addressed as factors in developing experimental protocols. Lastly, the erythrocyte perfusion uses a mix of bovine sources for red blood cells, an addition of the crystalline buffer, Krebs-Hanslett buffer, but also albumin as well as an oxygen-carrying protein. This results in a perfusate system, once again, that is able to maintain physiological osmolality as well as oncotic pressure. We also find that the hearts that are subjected to erythrocyte perfusion do maintain physiological perfusion rates for a much significantly longer period of time. However, once again, there's an incremental cost increase in this system, and it's an incredibly laborious procedure involved to actually make the perfusate.